So this is my presentation. It's by myself, Athena Krikorian, and I was mentored by Dr. Amity Manning. And let's just get into it. A little bit about my presentation. I'm going to be covering skin cancer and going into depth about really the malfunctions in the cell cycle as well as cell division, and diving into the specifics of cancer. And my interest and motivation for this actually came from my own personal interest in dermatology as a possible career for myself, and close relatives around me who had actually struggled with skin cancer. And I had heard lots of stories about people who had skin cancer. So that was kind of my motivation. And also, I tied this in with my passion for the climate crisis, our current climate crisis, and as well as the ozone layer depletion, which is basically our Earth's sunscreen, and it kind of protects us from everything. And while global warming is a big issue, the ozone layer is depleting because of it. And that's a big contributing factor to skin cancer cases rising. And my hopes for the future is to kind of get a closer understanding of the different proteins and genes in the cell cycle that contribute to the rapid cell division and tumor growth in all types of skin cancer. Okay, so some skin cancer forms, there are actually 3 types. And just as an introductory, my presentation is very much introductory based. And I kind of had a rudimentary knowledge of all of this at the beginning. I understood where I wanted to go with my research, but I wasn't as centered around understanding the different types of skin cancers, which is actually really interesting because cancer is cancer and it forms in specific ways. However, what I learned with my research with Dr. Manning is that there are actually specific malfunctions in the cell cycle that we will go into and explore. But the 3 main types of skin cancer is basal cell carcinoma, squamous cell carcinoma, and melanoma. And like I mentioned, the ozone layer depleting is a big cause of it, of many skin cancer cases going up, as well as strong UV light exposure over long periods of time from the sun. And indoor tanning beds are also like something that's huge in causing skin cancer. And not all cases are terminal. So there's definitely lots of therapies that I actually will go into at the end of my presentation. But the most kind of popular or effective types of treatment for skin cancer is actually chemotherapy, as well as stem cell research and stem cell therapy. Sorry. And so we're also going to explore where the skin cancer can start. And that is something that was really important to me as well, because we always hear about skin cancer being uncontrolled cell division, but we don't really understand how that happens. So many skin cancer cases are found to be linked to the RB1 gene, which is actually what my entire presentation is about. The RB1 gene and the PRB protein that I'm also going to explore and kind of explain, and how those two, the gene and the protein, can contribute to malfunctions in the cell cycle. So what is the RB gene? The RB1 gene. One of the main proteins of the cell cycle regulation is the RB1 gene. And PRB, the protein of this RB gene, is also what contributes to that regulation. And it stands for retinoblastoma because the RB1 gene was the first gene or malfunction found in retinoblastoma, which is a type of cancer and the type of eye cancer. And scientists discovered it, and they named it after the retinoblastoma cancer because that was the first time they discovered it, really. And so the RB1 gene regulates the rate at which cells divide in the cell cycle, and it also stops other proteins from triggering DNA replication in the case that it will not create a tumor. And this happens because of DNA replication, and because obviously it's a very basic biology concept where you have to do DNA replication before you go to before a cell can actually divide. And the RB1 gene is in charge of that, and it kind of regulates that. So if you don't have a proper DNA replication going on before a cell divides, many problems can happen. And also the RB1 gene manages cell survival, apoptosis, and differentiation, which is all important factors in contributing to cell growth and further on cell division. So how can the RB1 gene malfunction? There's actually 2 main ways, and they're polar opposites, but they both contribute to uncontrolled cell division. So the first one is mutations in the gene that become mutated, and they lead for it to create an incomplete or kind of inactive PRB protein. And that doesn't make it function normally at all, and it actually throws off cell division. So you could kind of imagine where there's a gene that's in charge of kind of regulating the cell cycle. If that continues to mutate, then the cell cycle will just continue to go on, and that division will continue to form basically, and it causes a tumor. And the second type is actually deletion in the gene, and that can also lead to uncontrolled cell division. And as you could tell, they're polar opposites. One is being deleted, one is being mutated. And also PRB binds with other proteins as well to make that clear. It's not just RB. And I'm going to kind of mention a few more of the different genes later in my presentation, but just to kind of get an idea of it, PRB is actually going to bind with other cellular proteins, and it's further going to kind of push out its growth and its growth suppression ability to bind to other proteins. And this is done by its phosphorylation state. And phosphorylation, basically, it's whenever it's like an addition of an amino group to a gene or a protein. And so when something is phosphorylated, it can't really bind to other proteins and it becomes inactive. So that's in hyperphosphorylation. And we have two different types of phosphorylation. One is hyper with the ER, and one is hypo with the O at the end. And their hypo is less phosphorylation, which is actually favored for the cell cycle, because we don't want our protein to be too heavily phosphorylated, or else it can become inactive and it won't be able to bind with other proteins. So both malfunctions, like I said, whether it's deleted or heavily phosphorylated, they still disrupt the cell cycle, and they still kind of add to that uncontrolled cell division. So for protein activity, like I mentioned, I would mention the further proteins that PRB binds with, and that is actually E2F. So PRB and E2F proteins are what work together to regulate the cell cycle. And PRB binds to E2F and it represses gene transcription. And this is kind of, it goes in increments, kind of at a manually rate. And again, this is actually the transcription factor, which is important for the DNA replication and cell division. Now for E2F, we see that it can actually promote the expression of genes, and that's with the genotype and how cells can continue on to express themselves. And they're required for the G1 to S phases in the cell cycle. So again, key factors that contribute to what's building up to our cell division. And this is kind of how I'm going to introduce how things could go really wrong. And as well as for PRB, when I mentioned that hypophosphorylated, we would rather prefer it to be hypophosphorylated because it could bind to E2F easily. And as long as E2F is bound to PRB in that hyperphosphorylation state, then it cannot promote gene transcription. And that's why the hyperphosphorylation is less favorable, because then the cell cannot enter the cell cycle. And additionally, when PRB is hypophosphorylated, E2F can't bind to PRB, and it causes E2F to kind of freely promote gene expression, and it allows the cell to actually enter the cell cycle. Now for phosphorylation enzymes, like I said, this is kind of going to be an introductory to everything that is involved with cell division and how the cell cycle is thrown off. And this is going to be a completely new concept, but just bear with me, because once I kind of introduce the CDK and cyclin-dependent kinase, which is a phosphorylation enzyme, everything will kind of come together. And it's actually the enzyme that puts phosphorylation marks on PRB. And whenever we talk about hypophosphorylation, which is what's favored, the CDK is what basically controls that. And it's what adds those amino groups, and it also regulates the different phases of the cell cycle. However, mitogens and CDK are actually partners. So mitogens tell the cell when to divide, and it activates CDK to add phosphorylation onto the cell. So it activates CDK proteins in normal cells, but in cancer cells, mitogens cannot really get to all the CDK proteins because there's such an abundance of them because of that uncontrolled cell division. And that's what happens whenever CDK is a lot more active without the mitogens present. And high levels of it in skin cancer mean that the PRB is going to be phosphorylated all the time. And so PRB won't ever get a chance to bind to E2F because it's so heavily phosphorylated. And that's wherever we, again, tie it back to being overly phosphorylated, where the hyperphosphorylation kicks in. It's whenever the CDK has been in so much abundance and the mitogens can't get to all of them. And growth with this becomes unregulated, like I mentioned, and CDK does not need the mitogens to kind of govern or control them anymore because there is so much, and it's kind of unattainable for the mitogens to get to all the different CDKs in the cell. And also dephosphorylation, which is really important, is done by a protein called PP1. And this is what actually takes phosphorylation marks off of RB. And so dephosphorylation is super important. It can happen naturally or with some therapies and drugs that will be mentioned later. And these are the antagonists to CDK. And they basically reverse and throw back all those phosphorylation marks. And that's done with the PP1 protein. So some skin cancer therapies, there's many, and we've been trying to, when I say we, I mean scientists and the world basically has been trying to prevent the tumor cells from entering the cell cycle again and stop that cell division from occurring, from going over the phosphorylation marks. And so certain therapies are actually reactivating PRB so that it can bind to E2F, even whenever it's been heavily phosphorylated. And we could tie this back to the dephosphorylation, where it could occur through therapies or naturally. And palbociclib is it's a drug that is an inhibitor of CDK activity, and it stops basically PRB from it stops basically CDK from phosphorylating PRB in order for the phosphatases to return to PRB and return it to its active, normal, hypophosphorylated state. And when we talk about phosphorylation, like I said, we mean the addition of amino groups to proteins, and hypo means less, hyper means more. So we're trying to go back to that hypo state where it's less and we're dephosphorylating. And PRB is also able to bind to E2F and inhibit those transcription factors from stopping those cancer cells from dividing. I also want to mention that as skin cancer kind of becomes a higher concern to people due to global warming and exposure to UV rays, and there's so many drugs and therapies being used, and even new research, we don't have a complete understanding. So the goal of my presentation and my entire literature review with the help of Dr. Manning was to get my kind of claws digged into what this could be. And I hope to explore this more in the future, maybe in more programs such as Talaria. So I'm really thankful for this opportunity. And thank you to my mentor, Dr. Manning. And I will now be taking questions if you have any.